Hello champions, welcome back to PU2 Brahmastra 2.0 series, and in this video we are going to discuss the most important questions from the chapter Biomolecules. So before starting the question, let us understand the mark distribution. So Biomolecules carries 9 marks, so it is even much more I can say higher weightage than your amines chapter, and this 9 marks is divided in this way. There will be one 5 marker question, one 2 marker question, so 5 plus 2 7, and there will be 2 1 marker question, that is 9 marks. Okay, now let's start with the first question. We'll go for this MCQ questions first. So the first question here, select the correct statement. Okay, we have to select the correct statement regarding amylopectin. Okay, so if you know what is amylopectin, amylopectin is a I can say is a part of the starch, right? Starch exists in 2 forms, amylose and amylopectin, and so we have been given statements regarding amylopectin. So amylopectin is a polymer of alpha D glucose units. Yes, that is correct. It constitutes of 15 to 20% of the starch. No, amylopectin consists of almost like 85 to 80%, so that is why this statement is incorrect. It is a water insoluble component. Yes, so we have 1 and 3 as the correct statement, which is option number A here. Moving to the second question, the number of amino acids in tetrapeptide. So in tetrapeptide there will be 4 amino acids, so that is why option number C here. Next, which of the following statements is not correct regarding the lactose? Okay, so lactose is a disaccharide of glucose and galactose. So on hydrolysis, lactose gives equal amount of glucose and galactose. That is a correct statement. And in lactose, there is a beta glucoside formed by the union of molecule of D, glucose and galactose. That is also correct. Lactose is a reducing sugar. That is also correct, and does not exhibit mutarotation. Lactose contains 7 OH groups. Okay, so if you see now, you have to find the incorrect statement. Next statement, lactose contains 7 OH groups. See, this statement is incorrect because lactose contains 8 OH groups. So option number D is the correct answer. Okay, let's see the next question. Okay, just a minute. Yeah, let's see the next question. Which of the following statements is not correct regarding the similarity between glucose and fructose? Both have pentaacetyl derivatives with acetic anhydride. With HCN, both give cyanohydrin. With phenylhydrazine, both give similar osazones. Both contain aldehyde functional group. This is the not similarity because fructose contain ketone functional group. So that is why option number D here. Next, in both DNA and RNA, heterocyclic base and phosphate ester linkages are at. So if you have in both DNA and RNA, okay, so if you have a nitrogenous base here, okay, if you have a nitrogenous base here, then on the, that is on this carbon, first carbon, so this is a sugar group. On the first carbon we have the base, okay, and then here we have 1 2 I can say 1 2 3 4 5, this carbon we have the phosphate group, right? So what I can say that the heterocyclic base and the phosphate ester linkages are, okay, the heterocyclic base is on the first carbon and the phosphate ester linkage is present on the 5th carbon, so that is option number C here. Next, select the incorrect statements from the following. The glucose exists in 2 different crystalline form, alpha and beta. That is a correct statement. Alpha and beta glucose are enantiomers. No, that is an incorrect statement. They are anomers. Okay. Next, what is there? Cellulose is a straight chain polysaccharide made up of only glucose units. Yes, that is a correct statement. Starch is a mixture of amylose and amylopectin. Both contain unbranched chain. No, amylopectin contains branched chain, so this is also incorrect. Okay, so what we have to find out? 1 and so what are my correct statements? 1 and 3, right? 1 and 2. Okay, select the incorrect statements from the following. Okay. Okay, incorrect statement. So we have 1 and 2 are incorrect, but not 3 and 4. No, 2 and 3 are incorrect. 2 is incorrect, definitely. 2 and 4 are incorrect, but not 1 and 2. Yes, 2 and 4 are incorrect, but not 1 and 2. That is option number C. Next, which of the following statements is incorrect? Again, proteins have a polyamide chain while nucleic acids contain polynucleotide chain. That is a correct statement. Next, we have nucleic acids are long chain polymers of nucleotides. That is also correct. RNA is a major source of genetic information which is copied into DNA molecules. No, DNA is the major source of genetic information. Proteins are synthesized in a process involving the translation of RNA. That is a correct statement. Next, which of the following groups includes all essential amino acids? Okay, so you have to see the list. This is the list of all essential amino acids. So in the first one, I have leucine, I have methionine, I have lysine, and we have valine. So yes, the first one itself contains all the essential amino acids. So option A is the correct answer. Okay, you can see in option number B, we have glycine, but glycine is not a essential amino acid. Similarly, in option number C, we have glutamine, which is not an essential amino acid. Then in this also you can see that we have tyrosine is there, which is not an essential amino acid. So you must remember these 1, 2, 3, 4, 5, 6, 7, these 10 essential amino acids you must remember. Okay. Next, the alpha and beta forms of the glucose they are anomers of glucose. So that is why option number C here. Okay, next we have thymine and pyridoxine are also respectively known as. So thymine is known as for vitamin B1 and pyridoxine is vitamin B6. So option number A is the correct answer here. Okay, now let's move to 2-marker question, because as I told you, there will be 2-marker question and 5-marker question in this section. So first one is what are reducing sugars and give example. So those sugars which can reduce tolerance reagent and Felling's reagent, or especially tolerance and Felling's reagent, are called as reducing sugar. So all the monosaccharides are your reducing sugars. So you have glucose, galactose, these are all reducing sugars. Next question, what are non-reducing sugars? Thus the opposite, the carbohydrates which cannot reduce tolerance and Felling's reagent are called as non-reducing sugars. Example here is sucrose. Next, 2 methods of preparation of glucose. So first of all, you can from sucrose. So if you take sucrose and then you hydrolyze it, you will get glucose and fructose. Next one is from starch. If you hydrolyze starch also, you will get n number of glucose molecules. Question number next one, how does glucose reacts with hydrogen iodide, or how do you show that glucose contains 6 carbon atoms in a straight chain? So for this one, we have it is like if you react glucose with HI, you get N-hexane, and this gives us the information that there are 6 carbon in the straight chain of a glucose molecule. Okay, so you can see glucose on heating with HI gives you N-hexane, so you have to write down this reaction. Next, how does glucose reacts with hydroxylamine, or how do you show that glucose contain a carbonyl group? So for presence of to show the presence of carbonyl group, it reacts with this NH2OH to give you oxime. Okay, so you can see here glucose reacts with hydroxylamine to give you glucose oxime. This shows that glucose contain a carbonyl group. Okay. Next question, how does glucose reacts with acetic anhydride, or how do you show that glucose has 5 OH groups? So when glucose reacts with acetic anhydride, all the OH groups, we get pentaacetyl derivative, which shows the presence of 5 OH groups. Okay, so you can see glucose and acetylation will give you pentaacetate, which confirms the presence of 5 OH groups. Next, how does glucose reacts with bromine water, or how do you show that glucose contains an aldehyde group? So glucose reacts with bromine water to give you gluconic acid. Okay, this is important, to give you gluconic acid, and this indicates the carbonyl group present in the glucose is an aldehyde group. Okay. Next, how do you show the presence of alcoholic group in glucose? Okay, so how do we show that? So for that, on oxidation with nitric acid, when this is there, you have to show that there is a presence of primary OH group. Okay, glucose as well as gluconic acid yield a dicarboxylic acid, and this indicates that there is a primary OH group present in the molecule. Okay, next you have to draw the open chain structure. So this is the formula of the open chain structure. Next, what are the limitations of open chain structure? The first one is it does not react with Skiff's reagent or with NaHSO3. The second one, the pentaacetyl derivative does not react with hydroxylamine. And then glucose exists in 2 different forms, that is alpha and beta form, which cannot be explained by the open chain structure. Next, what are epimers and what are anomers? See, when the sugars will differ in any one configuration, then they are called as epimers. Now, if the sugars which differ in their configuration at only C1 position or on the first carbon only, then it is called as anomers. Clear? Next, we have draw the Haworth projection of alpha D glucose and beta D glucose. So you need to remember, you need to practice this one, and remember, in alpha the OH group is at the down and in beta the OH group is present at the top. Okay. Next, we have write the Haworth projection of alpha D maltose. So we have drawn the structure of maltose. So you have to understand C1 C4 linkage is there between alpha D glucose. Next, mention the type of glycosidic linkage in maltose. This is your glycosidic. The type of linkage is your C1 of one glucose is linked with C4 of another glucose. And next, give reason why maltose is a reducing sugar. You can see here that you have one OH group and OR group, right? This OH group can be converted to free aldehyde group, and that is why this is an reducing sugar. The free aldehyde group can be produced at C1 of one of the glucose in solution, so it is a reducing sugar. Next, write the Haworth projection of lactose. So this structure you have to draw. Mention the type of glycosidic linkage. So the glycosidic linkage is C1 to C4, right? So we have C1 to C4 beta D galactose and C4 of beta D glucose. Next, reason why lactose is a reducing sugar. So here also you can see lactose has COH. This OH group can be converted to a free aldehyde group at C1 position. So that is why it is a reducing sugar. Next, name the storage polysaccharide present in animals. So it is glycogen, and where it is found, liver, muscles and brains. Next, we have two difference between starch and cellulose. So starch is made up of alpha D and this is made up of beta D. Here alpha glycosidic linkage, here we have beta glycosidic linkage. Here we have C1 C4 and C1 C6 linkage. Here we just have C1 C4. This is linear and branched, and here we have only linear polymer. Okay. Next, what are essential amino acids? Give an example. And what are non-essential amino acids? Give an example. So essential amino acids are those which cannot be synthesized in the body and can be and has to be consumed from the diet. Example, lysine, valine and leucine. Non-essential amino acids are that can be synthesized in the human body, glycine, alanine or serine. Next, question number 24, what are Zwitter ions or dipolar ions? So what happens, see, if you know the structure of the amino acid, okay, you have C, you have COOH, you have NH2, we have R, we have H, right? Now this one donates the H plus, okay, donates the H plus, and we get COO minus and NH3 plus, okay. So what is the structure formed here? The structure formed here is CH, COO minus, NH3 plus, and we have R group, right? So this group, this structure is a Zwitter ion structure, okay. So in aqueous solution, what happens? COOH group donates a proton and amino group accepts a proton, giving rise to a dipolar ion known as Zwitter ion. Next, how are proteins classified based on the structure or molecular shape? So proteins are classified into two shapes, that is fibrous and globular, okay, based on their structure. Fibrous and globular. Now what is fibrous proteins? They are fiber like structures and they are insoluble in water, okay. Next, in this the polypeptide chains run parallel and are held together by hydrogen or disulfide bonds. You have example keratin, myosin and collagen. Now what is globular proteins? They are spherical in shape and soluble in water. The spherical shape is due to the coiling of the polypeptide chains. Example, insulin, albumin and hemoglobin, okay. Next, what is denaturation of protein? Which level of structure remaining intact during denaturation of globular protein? Okay, so here we have the loss of, so when you have a protein and when you heat it, okay, when you just provide the heat to that, the loss of biological activity of protein by heating or the change in pH or presence of salts is called as denaturation, okay. Now after this, what happens? The protein structure is destroyed and it cannot function normally, and the primary structure, this during denaturation of protein, the primary structure is intact, okay. Next we have classification of vitamins. So we have fat soluble vitamins and water soluble vitamins. Fat soluble means it is soluble in fats and oils, insoluble in water. Example, we have A, D, E and K, and B and C is soluble in water. So that is why what happens, they are released from our body during sweating or urination. So that is why you need to take this B and C vitamins as a supplement. Okay. Next, the very important part from where majority of the questions will be asked, different types of questions will be asked, will be from the vitamins, their sources and their deficiency diseases. Not only in PU2, in KCET, Comet K, everywhere these questions are asked. So please prepare this table thoroughly, especially the vitamin and their deficiency diseases. So for example, vitamin A, the deficiency is xerophthalmia and night blindness. Okay, vitamin B1 is called as thiamine and its deficiency disease is beriberi. Vitamin B2 is called as riboflavin and its deficiency disease is cirrhosis. Next we have vitamin B6, which is called as pyridoxine and it causes convulsions. Next we have vitamin B12, which has pernicious anemia. Okay, means where the RBC cells, the shape of the RBC changes. Next we have vitamin C, ascorbic acid, which causes scurvy. Next we have vitamin D, which causes rickets. Next we have the differences between DNA and RNA. So we have double-stranded DNA and in RNA we have single-stranded nucleotide chains. Okay. Now what is the sugar used in DNA? The sugar here used is deoxyribose sugar. Okay. And the nitrogenous bases here are A, T, G and C. And next, what is the sugar used here? It is ribose sugar. Okay. And we have A, U, G and C here. Clear? Okay, next we'll talk about hormones. So in general, there are I have not given you direct questions from hormones, but I want to discuss, because if you see in textbook, the hormones part is very scattered. It is not given in a proper way to understand. So I have made you proper notes and I'll explain you each and everything so that you can study from here directly. Okay, so let's see. First one is, what is hormones? They are intracellular messengers, means they are directly poured into the bloodstream. Okay, and they are secreted by endocrine glands. Now they are poured directly. And what are the types of hormones we have? We have first of all polypeptide, amino acid derivative and steroids. Okay, these are three categories of I'll say hormones we have. Now in polypeptide we have insulin, endomorphins and glucagons, and in amino acid derivatives we have epinephrine, norepinephrine and thyroxine. Okay, epinephrine, norepinephrine and we have thyroxine. Then in steroids we have estrogens and androgens. Okay, so we'll see each of them in detail. So first of all, if you see glucagon is a polypeptide. Okay, I'll write here. So we have major three types of hormones: polypeptide, amino acid derivatives and we have steroids, right? So where do we have glucagon? Glucagon is a polypeptide. Okay. Now what does it cause it? It increases the sugar level in the blood. And if you see insulin is also a polypeptide, right? Insulin is also a polypeptide. Now insulin works to reduce the sugar level in the blood. So these two, glucagon and insulin together, maintains the sugar level in the blood. Okay. Now coming to epinephrine and norepinephrine, these are amino acid derivatives. Okay, amino acid derivatives. Okay, epinephrine and norepinephrine. Now here their responses or their function is the response to external stimuli. Okay, external stimulus. Next we'll talk about thyroxine. Thyroxine is also amino acid derivative. Thyroxine is also amino acid derivative. Okay, now here what happens, they are produced in the thyroid glands. Okay, they are produced in thyroid glands. Next we'll talk about what is this thyroxine? This is also asked many times in the question. Thyroxine is the iodinated derivative of tyrosine. Tyrosine is an amino acid. So if one of that it is replaced with iodine, we get thyroxine. Okay. Next, if there is low concentration of thyroxine, we suffer from hypothyroidism, or it is called and which leads to lethargies and obesity. And on high concentration of thyroxine, we suffer from hyperthyroidism, which is your enlargement of the thyroid gland. You might have seen people having goiter, right? So that is because of this thing. Next we will talk about steroid hormones. Okay, so steroid hormones are of again two types. The steroid hormones are produced by two important I can say organ or parts, that is adrenal cortex and gonads. Okay, now adrenal cortex, so glucocorticoids, this is a steroid hormone produced by adrenal cortex. Okay, what is the function of glucocorticoids? They control the carbohydrate metabolism, modulate the inflammatory reactions, and are involved in the reactions to stress. Okay. Next we have mineral corticoids. They control the level of excretion of water and salt by the kidney. Okay. So next we have Addison's disease. So what is this Addison disease? This is when you have the malfunctioning of adrenal cortex. Now see, if adrenal cortex is malfunctioning, then your glucocorticoids and mineral corticoids will not be synthesized properly. So what will be the symptoms of that? You will have hypoglycemia, weakness, and increased susceptibility to stress. Now why will there be increased susceptibility to stress? So if you see the function of glucocorticoids, it has functions in the reactions to stress. Now if that is not produced properly, that is not synthesized properly, then your body will have abnormal reactions to stress conditions. Okay. Next we'll see the hormones, that is the hormones that is released by, or I can say not hormones, I'll see the steroid hormones released by gonads. Okay, so there were two things, right? Adrenal cortex and gonads. So we have understood glucocorticoids, mineral corticoids. So these are the steroid hormones released by adrenal cortex. Now which I am discussing, that is testosterone, estradiol, progesterone. These are the steroid hormones released by gonads. Okay, so here if you see, they are the sex hormones present in the males. Testosterone, okay, they are responsible for development of secondary male characteristics like deep voice, facial hair, general physical constitution, etc. Now estradiol is the female sex hormone, and it is used in the development of secondary female characteristics and the control of menstrual cycle. Okay, next progesterone, it is also a female sex hormone. It is responsible for preparing the uterus for the implantation of the fertilized egg. Clear this one? Okay. Next, we will talk about a long answer question. So already whatever 2-marker questions I have discussed, right? If you just club them, you know there is a 3+2 system, right? So there will be mix and match, and you will be getting 5-marker questions. Okay. So I have not taken specifically more 5-marker question. I have taken more 2-marker question. If you do them by mix and match, you will be able to answer all the 5-marker questions. Okay, so let us see the first one, the Harvard structure of sucrose. So you can see this is the Harvard structure of sucrose. Give reason that sucrose is a non-reducing sugar. So you can see that in sucrose there is no free aldehyde group. Okay, reducing groups of glucose and fructose are involved in glycosidic bond. Sucrose is a non-reducing sugar because there is no free aldehyde group. Next, mention the glycosidic linkage in sucrose or cane sugar. So it is from C1 of alpha D glucose and C2 of beta D fructose. Next, mention the glycosidic linkage. This is done. What is invert sugar? Now see, we have understood when I have discussed the preparation of glucose, right? We have studied that sucrose can undergo hydrolysis to give you glucose and fructose. Okay. Now sucrose is dextrorotatory. Dextrorotatory means it will rotate the plane polarized light in clockwise direction. Now you have taken sucrose and you added some acid, so hydrolysis will take place. And once the hydrolysis started, we have glucose and fructose in the sample, right? Now when you pass the plane polarized light, it will have levo-rotation. Okay, now why that is happening? Because there is glucose and fructose in that. So glucose is having dextrorotatory, but the fructose is having more levo-rotation than glucose. Okay, so after cancellation, the net effect comes out to be the anti-clockwise direction. So now see, the sample was there, you passed plane polarized light initially, it was dextrorotatory. After certain interval you pass the plane polarized light, it is levorotatory. So that is what the inversion happens here. Okay, so you can see here sucrose is dextrorotatory, but after hydrolysis it gives you dextrorotatory glucose and levorotatory fructose. Okay, since the levorotation of fructose, that is minus 92 degree, is more than that of the dextrorotation of glucose, the mixture is levorotatory, right? So you can see up to plus 52 minus 52 will cancel. The next what is rest is, that is your anti-clockwise direction. Thus hydrolysis of sucrose brings about a change in the sign of rotation from dextro to levo, and that is why it is named as invert sugar. Okay. Next is, what is a glycosidic bond? So when two monosaccharides are joined by, you can see, are joined together by an oxide linkage formed by the loss of water molecule. Such a linkage between two monosaccharide units through oxygen atom is called as glycosidic linkage. Okay. Next we will see question number 2. Name the storage polysaccharide present in the plants. So the storage polysaccharide present in the plants is your cellulose. Right? Sorry, just a minute. Okay. Storage, it is your starch. Okay. Next, name the monomer units present in starch. It is your alpha D glucose units. Okay. Name the components of starch. So we have amylose and amylopectin. Okay, these are the components of starch. Then write the two difference between amylose and amylopectin. So this is a very potential question. So amylose is water soluble, this is water insoluble. 15 to 20%, this is 80 to 85%. Unbranched, this is branched. Linkage is C1 to C4 between alpha D glucose units. Here it is C1 to C4 between alpha D glucose units and C1 to C6 between the branching molecules. Okay. Okay, next I want to do a quick revision of all the monomer units and everything so that you can learn it very easily. So see, in disaccharides we have 3 disaccharides: sucrose, maltose and lactose. In sucrose, what is the connection? Alpha D glucose and beta D fructose. These are the monomer units. What is the linkage? C1 of alpha D glucose and C2 of beta D fructose. Next, maltose is alpha D glucose. Okay, only alpha D glucose is there. C1 of glucose and C4 of another glucose. Next, we have beta D glucose and beta D galactose. So it is C1 of beta galactose and C4 of glucose. Okay. Next, we have polysaccharides. In that we have starch, and starch comprises of amylose and amylopectin. Then we have cellulose, and then we have glycogen. So starch is your alpha 1,4 glycosidic bonds and amylopectin is alpha 1,4 glycosidic bonds with 1,6 branches. Okay, there will be branching at this position. Cellulose is beta 1,4 glycosidic bonds. Glycogen is alpha 1,4 glycosidic bonds with or alpha 1,6 branches. Okay, so it is same as amylopectin, but it is further more branched. It is more branched than amylopectin. Okay, next we'll talk about some important topics that you must prepare from this chapter. So first of all, glucose, all the 6 reactions which tells about the structure of glucose. The first one, that is reaction with HI. The second one, that is the reaction with NH2OH. The third one, reaction with bromine water. The fourth one, reaction with anhydrides. And the fifth one, that is the reaction with your nitric acid. Okay, so this all, what are the reactions, what are their conclusions, you should remember, because from here questions will be definitely asked. Next, disaccharides and polysaccharides, their examples, and this, especially this chart you have to remember, where you have to know their monomer units, their diagrams and the connectivity. Next is reducing sugars and non-reducing sugars, definition and examples. So this is all about carbohydrates. In proteins, you should know about structure of protein, primary, secondary, tertiary structures. Then peptide bond formation, Zwitter ion, and what is essential and non-essential amino acids. One more thing I want to add here, which I missed, is denaturation of protein. It is also a very important topic, denaturation of proteins with examples. Okay, next for vitamins deficiency diseases is very important. Okay, it will be asked either in MCQ, in the fill in the blanks, match the following. Every year deficiency disease questions are asked, not only in this KCET, Comet K, JEE, every exam has this question, so please prepare it thoroughly. Okay. Then for new hormones, you should know all the functions of all the hormones. Okay, I have given you the table. Please study from here only, because in book it is very cluttered. You will not find the proper, so I have given you proper category, like what we have the amino acid derivatives, then we have the steroid hormones, right? And then from each category, what are the hormones present and what are their functions present. Okay. Next we have nucleic acids, RNA DNA difference, and then you should know the structure. DNA difference in terms of the sugar that is used, the bases that is present, the strands they have, that is important. Then what is nucleic acids, what is nucleoside, what is nucleotide, how the composition happens, where the bond is joined. So these all things you should be revising. Okay, so these are the important topics, and with this we are, okay, one second. Yeah, and with this we have completed this chapter, all the important questions, topics, everything I have told you in this video. So please watch it completely and follow it seriously. See, I know this biomolecules chapter, many students like who are in computer science, they don't study, right? But let me tell you, this is something which is very factual. You just need to make proper notes and just revise the notes, like I'm telling you, for this particular chapter, understanding is less required, more of rote memory is required, because there are so many names, so many factors, so many, like there's less use of concept but more use of rote memory. So please make proper notes, and before the exam you just mug it up and just solve questions from this. Okay, that's the way how to tackle this chapter. Please make proper notes from this. Okay, thank you so much and all the best, and I hope you would have liked this session. Still, if you have any doubts, put down in the comment section. I'll definitely address it.